Why don't we get to the data on the IL-23 blockers, of which there is a ton of data. Who wants to tell us about the reSURFACE data with tildrakizumab?
I could take
Scott,
that
go
one.
ahead.
So, so there were two trials done, reSURFACE 1 and reSURFACE 2. One trial, they were basically one-year trials. The primary endpoint occurred at week 12, and that was PASI 75. The dosing, of course, is 100 milligrams at week 0, 4, and every 12 weeks thereafter. The actual clinical trials had a more robust study design with other dosing schedules, but the FDA-approved doses at week 0, 4, and every 12 weeks thereafter. And all primary endpoints were met. So, roughly around 2/3 of patients achieved a PASI 75 at week 12. But what's interesting about tildrakizumab, and I think is important for people to understand, is that that's an example of a drug where the primary endpoint is by far not where you see maximum efficacy. So they got dosed at week 0 and 4, came back at week 12 for their primary endpoint evaluation, and then they got a third dose at week 16. They came back at week 28 for a secondary evaluation, and they did much better than at week 12. So by week 16, PASI 75 was around 75% is sort of how I gestalted it in my mind. The same thing was true of PASI 90. So patients, about a third of patients did well, had achieved PASI 90 at week 12, but with that third dose, that went up to close to half of the patients. And similarly, PASI 100 had a sort of modest response at week 12, but a much more robust response. About a quarter of the patients or so achieved PASI 100 at week 28. Furthermore, in that study, if you are a PASI 75 responder at week 28, then you then you were able to go into the extension trial, and those patients who continued on drug did well. So about 85% of patients who were PASI 75 responders at week 28 maintained PASI 75 out to week 52 or 64. In addition, there's some long-term data so that if you stayed on the drug out to 3 years, you've maintained efficacy. So I think we have nice efficacy data that it works. It may not be the quickest one out of the block, but I don't think that's my goal. My goal is to, win the race, or at least do as good as anybody else in the marathon.
Unless they're
I'm
getting
not a
married.
100-yard dash kind of guy. And my patients aren't either. They want to get better quicker, but as long as they see steady improvement, they're willing to wait. The nice thing about the drug is that you could tell very early on, by week 4, certainly by week 8, if your patient will do well long term. So if your patient has sort of a modest response, maybe PASI 40 or less at week 8, they're probably not going to be good long-term responders. But if they have a more robust response, maybe PASI 50 or 60 at week 8, you could tell that they will be a good long-term responder. So in my clinical practice, I give a dose at week 4, give a dose at week 0, give a dose at week 4, and then I might see them back at week 8 or 12 or whatever, even before their third dose. And if they're not doing well, I stop and I go to something else. But if they are doing well, then I know even if they haven't achieved, a PASI 75 or, a significant improvement at that point, I know that they will by week 28. And then the nice thing is if they do respond by week 28, then I know that they're going to continue to respond out to a year and possibly to 3 years. I think the safety profile of IL-23s as a class, and I would include Tildra, certainly among these, I mean, I don't see that there's virtually no signal. There's certainly things in the importance in the ISI part of the package insert, but I think that they're very reasonable. It mentions infection, it mentions TB, although there were no cases of reactivation of TB, and there were several dozen patients with latent TB in that clinical trial. It mentions not giving live vaccines. I don't know, Mark, maybe you could back me up on this. Has there ever been a patient who's been given a live vaccine who's developed the disease state for which they were given the vaccine?
No, you know that the best study on that was done for TNF blockers, which are actually substantially more immunosuppressive. Yeah. And what they showed, it was published in JAMA, yeah. And they divided the patients in two groups, patients on TNF blockers who got live vaccines and patients not on TNF blockers who got live vaccines. There were no more cases of, the vaccine not working. So the frequency of herpes zoster, which was the vaccine that was used, was no higher in the patients on TNF blockers. So the vaccine worked well. Right. And there were no cases of disseminated zoster. Right. And so, everything we were worried about did not happen. Nonetheless, the package insert does make the statement about live vaccines. I know that when we send our patients to infectious disease experts, they respect that. They actually are very wary about giving live vaccines to patients on any of the biologics. But I would agree with you that that is not evidence-based.